Good morning, this is Dr. Will, CBMD, PhD. This is Monday, October 24, 2011, 11:49 p.m. Eastern Time. Quick note, solving the mysteries of short-legged Neanderthals. Science Daily, October 19, 2011. While most studies have concluded that a cold climate led to the short, lower legs typical of Neanderthals, researchers at the Johns Hopkins have found that lower leg length, shorter than typical modern humans, let them move more efficiently over the mountainous terrain where they live. The findings reveal a broader trend relating shorter lower leg lengths to mountainous environments that may help explain the limb proportions of many different animals. And good morning, this is Dr. Will, CBMD, PhD. This is Monday, October 24, 2011, 11:52 p.m. Eastern Time. Quick note, studies looking at limb length have always concluded that a shorter limb, including in Neanderthals, leads to less efficiency of movement because they had to take more steps to go a given distance. And good morning, this is Dr. Will, CBMD, PhD. This is Monday, October 24, 2011, 11:53 p.m. Eastern Time. Quick note, Neanderthals who lived from 40,000 to 200,000 years ago in Europe and Western Asia, mostly during very cold periods, had a smaller stature and shorter lower leg lengths than modern humans because mammals in cold areas tend to be more compact with a smaller surface area. Scientists have normally concluded that it was the region's temperature that led to their truncated limbs compared to those of modern humans, who lived in a warmer environment overall. Good morning, this is Dr. Will, CBMD, PhD. It is Monday, October 24, 2011, 11:55 p.m. Eastern Time. Quick note, spiral arms hint at presence of planets. High-resolution image of young star with circumstellar disk verifies predictions. Science Daily, October 19, 2011. A new image of the disk of gas and dust around a sun-like star has spiral arm-like structures. These features may provide clues to the presence of embedded but as yet unseen planets. And good morning, this is Dr. Will, CBMD, PhD. This is Monday, October 24, 2011, 11:56 p.m. Eastern Time. Quick note, detailed computer simulations have shown us that the gravitational pull of a planet inside a circumstellar disk can perturb gas and dust, creating spiral arms. Now, for the first time, we are seeing these features. And good morning, this is Dr. Will, CBMD, PhD. This is Monday, October 24, 2011, 11:57 p.m. Eastern Time. Quick note, the newly imaged disk surrounds SAO 206462, a star located about 456 light-years away in the constellation Lupus. Astronomers estimate that the system is only about 9 million years old. The gas-rich disk spans some 14 billion miles, which is more than twice the size of Pluto's orbit in our own solar system. And good morning, this is Dr. Will, CBMD, PhD. This is Monday, October 24, 2011, 11:59 p.m. Eastern Time. Quick note, what we are finding is that once these systems reach ages of a few million years old, their disks begin to show a wealth of structure, rings, divots, gaps, and now spiral features. And good morning, this is Dr. Will, CBMD, PhD. This is Tuesday, October 25, 2011, 12:05 a.m. Eastern Time. Quick note, NASA telescopes help solve ancient supernova mystery. Science Daily, October 24, 2011, the mystery that began nearly 2,000 years ago when Chinese astronomers witnessed what would turn out to be an exploding star in the sky has been solved. New infrared observations from NASA's Spicer Space Telescope and Wide Field Infrared Survey Explorer WISE reveal how the first supernova ever recorded occurred and how its shattered remains ultimately spread out to great distances. And good morning, this is Dr. Will, CBMD, PhD. This is Tuesday, October 25, 2011, 12:07 a.m. Eastern Time. Quick note, the findings show that the stellar explosion took place in a hollowed-out cavity, allowing material expelled by the star to travel much faster and farther than it would have otherwise. And good morning, this is Dr. Will, CBMD, PhD. This is 185 AD. Chinese astronomers noted a guest star that mysteriously appeared in the sky and stayed for about 8 months. By the 1960s, scientists had determined that the mysterious object was the first documented supernova. Later, they pinpointed RCW 86 as a supernova remnant located about 8,000 years away, but the puzzle persisted. The star's spherical remains are larger than expected. If they could be seen in the sky today in infrared light, they'd take up more space than our full moon. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25, 2011, 12:12 a.m. Eastern Time. Quick note, Fire Volcano offers geological glimpse into the land that time forgot. Science Daily, October 19, 2011. First scientist to witness exploding rock and molten lava from a deep-sea volcano seen during 2009 expedition report that the eruption was near a tear in Earth's crust that is mimicking the birth of a subduction zone. And good morning, this is Dr. Will, CBMD, PhD. This is Tuesday, October 25, 2011, 12:23 a.m. Eastern Time. Quick note, followed a giant meteorite strike revealed a new model. Science Daily, October 19, 2011. Seeking to better understand the level of death and destruction that would result from a large meteorite striking Earth, Princeton University researchers have developed a new model that can now only more accurately simulate the seismic fallout of such an impact, but also help reveal new information about the surface and interior of planets based on past collisions. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 25, 2011, 12:24 a.m. Eastern Time. Quick note. Princeton researchers created the first model to take into account Earth's elliptical shape, surface features, and ocean depths in simulations of how seismic waves generated by a meteorite collision would spread across and within the planet. Current projections rely on models of a featureless, spherical world with nothing to disrupt the meteorite impact. The researchers report in the October issue of the Geophysical Journal of International. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 25, 2011, 12:27 a.m. Eastern Time. Quick note. The Princeton simulations also could help researchers gain insight into the unseen surface and interior details of other planets and moons. The authors reported the simulations can pinpoint the strength of the meteorite's antipodal focus, the area of the globe opposite of the crater where the energy from the initial collision comes together like a second, smaller impact. The researchers found this point is determined by how the features and composition of the smitten orb direct and absorb the seismic waves. Scientists could identify the planet or a moon's characteristic by comparing a crater of the remnants of the antipodal point and calculating how the impact waves spread. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 25, 2011, 12:29 a.m. Eastern Time. Quick note. We have developed the first model to account for how Earth's surface features and shape would influence the spread of seismic activity following a meteorite impact. For the Earth, these calculations are usually made using a smooth, perfect sphere model, but we found that the surface features of a planet or a moon have a huge effect on the aftershock of a large meteorite will have. So it's extremely important to take those into account. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 25, 2011, 4:10 p.m. Eastern Time. Quick note. Design rules will enable scientists to use DNA to build nanomaterials with desired properties. Science Daily, October 13, 2011. Nature is a master builder. Using a bottom-up approach, nature takes tiny atoms and, through chemical bonding, makes crystalline materials like diamonds, silicone, and even table salt. In all of them, the properties of the crystals depends upon the type and arrangement of atoms within the crystalline lattice. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25, 2011, 4:11 p.m. Eastern Time. Quick note. Now a team of Northwestern University scientists has learned how to top nature by building crystalline materials from nanoparticles and DNA, the same material that defines the genetic code for all living organisms. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25, 2011, 4:12 p.m. Eastern Time. Quick note. Using nanoparticles as atoms and DNA as bonds, the scientists have learned how to create crystals with the particles arranged in the same types of atomic lattice configurations as some found in nature. But they also have built completely new structures that have no naturally occurring mineral counterpart. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25, 2011, 4:13 p.m. Eastern Time. Quick note. The basic design rules the Northwestern scientists have established for this approach to nanoparticle assembly promise the possibility of creating a variety of new materials that could be useful in catalysts, electronics, optics, biomedicine, and energy generation, storage, and conversion technologies. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25, 2011, 4:15 p.m. Eastern Time. Quick note. We have developed modes of controlled crystallization that are, in many respects, more powerful than the way nature and chemists makes crystalline materials from atoms. By controlling the size, shape, type, and location of nanoparticles within a given lattice, we can make completely new materials and arrangements of particles, not just what nature dictates. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25, 2011, 4:17 p.m. Eastern Time. Quick note. In the study, the researchers start with two solutions of nanoparticles coated with single-stranded DNA. They then add DNA strands that bind to these DNA functionalized particles, which then present a large number of DNA sticky ends. At controlled distance from the particle surface, these sticky ends then bind to the sticky ends of adjacent particles, forming a macroscopic arrangement of nanoparticles. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25, 2011, 4:18 p.m. Eastern Time. Quick note. Different crystal structures are achieved by using different combinations of nanoparticles, with varying sizes and DNA linker strands, with controllable lengths. After a process of mixing and heating, the assembled particles transition from an initially disordered state to one where every particle is precisely located according to a crystal lattice structure. The process is analogous to how ordered atomic crystals are formed. Good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 4:20 p.m. Eastern Time. Quick note. The researchers report 6 design rules that can be used to predict the relative stability of different structures for a given set of nanoparticle sizes and DNA lengths. In the paper, they use these rules to prepare 41 different crystals structures with 9 distinct crystal symmetries. However, the design rules outline a strategy to independently adjust each of the relevant crystallographic parameters, including particle size varied from 5 to 60 nanometers, crystal symmetry and lattice parameters which can range from 20 to 150 nanometers. This means that these 41 crystals are just a small example of the near infinite number of lattices that could be created using different nanoparticles and DNA strands. Good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 4:25 p.m. Eastern Time. Quick note. National Geographic engineers deployed untethered free-falling ascending landers, equipped with digital video and lights to search the largely unexplored region. The team documented the deepest known existence of xenophyophores, single-celled animals exclusively found in deep-sea environments. Xenophyophores are not worthy for their size, with individual cells often exceeding 10 centimeters, 4 inches, their extreme abundance on the seafloor, and their role as host for a variety of organisms. Good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 4:29 p.m. Eastern Time. Quick note. As one of very few taxa found exclusively in deep sea, the xenophyophores are emblematic of what the deep sea offers. They are fascinating giants that are highly adapted to extreme conditions, but at the same time are very fragile and poorly studied. These and many other structurally important organisms in the deep sea need our stewardship as human activities move to deeper waters. Good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 4:37 p.m. Eastern Time. Quick note. Researchers build transparent, super stretchy skin-like sensor. Science Daily, October 24, 2011. Imagine having a skin so supple you could stretch it out to more than twice its normal length in any direction. Repeatedly, yet it would always snap back, completely wrinkle-free when you let go of it. You would certainly never need Botox. Good morning, it's Dr. Will, CBMD, PhD. It is Tuesday, October 25th, 2011, 4:38 p.m. Eastern Time. Quick note. That inviolable elasticity is one of several few features built into a new transparent skin-like pressure sensor that is the latest sensor developed. Good morning, this is Dr. Will, CBMD, PhD. This is Tuesday, October 25th, 2011, 4:39 p.m. Eastern Time. Quick note. This sensor can register pressure ranging from a firm pinch between your thumb and forefinger to twice the pressure exerted by an elephant standing on one foot. Good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 4:41 p.m. Eastern Time. Quick note. The key element of the new sensor is the transparent film of carbon nanosprings, which is created by spraying nanotubes in a liquid suspension onto a thin layer of silicone, which is then stretched. When the nanotubes are airbrushed onto the silicone, they tend to land in randomly orientated little clumps. When the silicone is stretched, some of the nanotube bundles get pulled into alignment into the direction of the stretching. When the silicone is released, it rebounds back to its original dimensions, but the nanotubes buckle and form little nanostructures that look like springs. After we have done this kind of pre-stretching to the nanotubes, they behave like springs and can be stretched again and again without any permanent changes in shape. Good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 4:46 p.m. Eastern Time. Quick note. Stretching the nanotube-coated silicone a second time in the direction perpendicular to the first direction causes some of the other nanotube bundles to align in the second direction. That makes the sensor completely stretchable in all directions, with total rebounding afterward. Additionally, after the initial stretching to produce the nano springs, repeated stretching below the length of the initial stretch does not change the electrical conductivity. Significantly, maintaining the same conductivity in both the stretched and unstretched forms is important because the sensors detect and measure the force being applied to them through these spring-like nanostructures, which serve as electrodes. Good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 4:49 p.m. Eastern Time. Quick note. The middle layer of silicone stores electrical charge, much like a battery. When pressure is exerted on the sensor, the middle layer of silicone compresses, which alters the amount of electrical charge it can store. The change is detected by the two films of carbon nanotubes, which act like a positive and negative terminals on a typical automobile or flashlight battery. And good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 4:55 p.m. Eastern Time. Quick note. Junk DNA defines differences between humans and chimps. Science Daily, October 25th, 2011. For years, scientists believed the vast phenotype differences between humans and chimpanzees would be easily explained. The two species must have significantly different genetic makeups. However, when their genomes were later sequenced, researchers were surprised to learn that the DNA sequences of humans and chimpanzees genes are nearly identical. What then is responsible for the many morphological and behavioral differences between the two species? And good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 4:57 p.m. Eastern Time. Quick note. Our findings are generally consistent with the notion that the morphological and behavioral differences between humans and chimpanzees are predominantly due to differences in the regulation of genes rather than to differences in the sequence of the genes themselves. And good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 5:22 p.m. Eastern Time. Quick note. Various phosphate-containing compounds, in which every phosphorus atom is the radioisotope phosphorus-32, are readily available. Because of their high specific activity, 32P labeled nucleotides are routinely used to label nucleic acids in cell-free systems, as discussed in the next section. However, phosphorus-32 is not suitable for certain types of incorporation experiments designed to localize labeled materials within the cell. The radioisotope iodine-125, which also is available in almost pure form, can be covalently linked to a protein or nucleic acid to yield preparations with a high specific activity. Such attachment of iodine-125 can be achieved enzymatically or chemically, and generally does not drastically affect the properties of a macromolecule. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 5:24 p.m. Eastern Time. Quick note. Radio-labeled molecules can be detected by visual and quantitative methods. Depending on the nature of an experiment, labeled compounds are detected by audio-radiography, a semi-quantitative visual assay, or their radioactivity is determined in an appropriate counter, a highly quantitative assay that permits the concentration of a radio-labeled compound to be estimated. In some experiments, both types of detection are used. Thanks. Good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 5:25 p.m. Eastern Time. Quick note. Audio-radiography and audio-radiography, a cell or cell constituent is labeled with a radioactive compound and then overlaid with a photographic emulsion, sensitive to radiation. Development of the emulsion yields smaller silver grains whose distribution corresponds to that of the radioactive material. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 5:29 p.m. Eastern Time. Quick note. Quantitative determination of radio-labeled compounds. The amount of radioactivity in a labeled material can be measured with either of two different instruments, Geiger counter or scintillation counter. The former detects ions produced in a gas by B particles or X-ray emitted from a radioisotope. In a later radio-labeled sample is mixed with a liquid containing a fluorescent compound that emits a flash of light. When it absorbs the energy of B particles or gamma rays released during decay of the radioisotope, a phototube, and the instrument detects and counts these light flashes. And good evening, this is Dr. Will, CBMD, PhD. This is Tuesday, October 25th, 2011, 5:31 p.m. Eastern Time. Quick note. In the usual experimental design, a radioactive precursor is incubated with whole cells or cell-free extracts. The cellular constituents then are isolated and purified in various ways. Finally, the radioactivity of the various fractions is measured with a counter. For example, to identify the site of RNA synthesis, cells can be incubated for a short period with 3H uridine and then subjected to a fractionation procedure to separate the various organelles. Chapter 5, the specific activity of the nuclear fraction. DPMMG protein is found to be much higher than that of any other organelle fraction, thus confirming the nucleus as a site of RNA synthesis. And good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 5:33 p.m. Eastern Time. Quick note. A combination of labeling and biochemical techniques and of visual and quantitative detection methods are often employed. For instance, to identify the major proteins synthesized by a particular cell type, a sample of the cell is incubated with a radioactive amino acid, 35S labeled methionine, for a few minutes. The mixture of cellular proteins then is resolved by gel electrophoresis, and the gel is subjected to an audio-radiographic radioactive band, correspond to newly synthesized proteins, which have incorporated the radio-labeled amino acid. Alternatively, the proteins can be resolved by liquid chromatography, and the radioactivity in the eluted fractions determined quantitatively with a counter. And good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 5:34 p.m. Eastern Time. Quick note. Intercellular precursor pools affect the outcome of pulse chase experiments. The purpose of some studies is to label the macromolecule and then trace the labeled compound as it moves through various cellular compartments, or is incorporated into other molecules within cells. This can be accomplished with a pulse chase experiment, in which cells are incubated briefly with a radio-labeled precursor, the pulse followed by removal and replacement of the labeled precursor by an excessive and unlabeled precursor. The chase. The fate of the newly synthesized compound that incorporated into the label during the pulse is tracked by measuring the radioactivity of the cells or cell fractions at various times thereafter. And good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 6:32 p.m. Eastern Time. Quick note. One of the most widely known features of a hypnotized person in the popular culture is a glazed, wide-open look and the eyes. Paradoxically, this sign has not been considered to have any major importance among researchers and has never been studied in any detail, probably due to the fact that it can be seen in only some hypnotized people. And good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 6:34 p.m. Eastern Time. Quick note. Published in the online journal of PLOS ONE, the study was done with a very high hypnotized participant who can be hypnotized and dehypnotized by just using one word cue. The change between hypnotic state and normal state can thus be varied in seconds. And good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 6:35 p.m. Eastern Time. Quick note. The researchers used high-resolution eye tracking methodology and presented a set of well-established ocular motor tasks that trigger automatic eye behavior. They found that glazed stare was accompanied by objectively measurable changes in automatic reflexive eye behavior that could not be imitated by non-hypnotized participants. And good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 6:36 p.m. Eastern Time. Quick note. In the field of hypnosis research, this result means that hypnosis can no longer be regarded as mental imagery that takes place during a total normal waking state of consciousness. On the other hand, the results may have wider consequences for psychology and cognitive neuroscience, since it provides the first evidence of the existence of a conscious state in humans that has previously not been scientifically confirmed. And good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 25th, 2011, 6:38 p.m. Eastern Time. Quick note. Hypnosis has had a long and controversial history in psychology and psychiatry and neurology for over 100 years. Researchers have debated if a specific hypnotic state exists or whether it is just about using cognitive strategies and mental imagery in a normal waking state. So far, a hypnotic state has never been convincingly demonstrated, and therefore many researchers regard the hypnotic state to be just a popular myth in psychology. Good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 26, 2011, 1:22 a.m. Eastern Time. Quick note. Watching motion of electrons and molecules during chemical reactions. Science Daily, October 14, 2011. A research group led by ETH now for the first time visualized the motion of electrons during a chemical reaction. The new findings in the experiment are of fundamental importance for photochemistry and could also assist the design of more efficient solar cells. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 26, 2011, 1:24 a.m. Eastern Time. Quick note. The research team irradiated nitrogen dioxide molecules NO2 with very short ultraviolet pulse. Subsequently, the molecule takes up energy from the pulse, which sets the electrons in motion. The electrons start rearranging themselves, which causes the electron cloud to oscillate between two different shapes for a very short time before the molecule starts to vibrate and eventually decomposes into nitric oxide in an oxygen atom. And good morning, this is Dr. Will, CBMD, PhD. It's Wednesday, October 26, 2011, 1:26 a.m. Eastern Time. Quick note. Nitrogen dioxide has model character with respect to understanding electron motion. In the NO2 molecule has states of the electrons can have the same energy for a particular geometry, commonly described as conical intersection. The conical intersection is very important for photochemistry, and frequency occurs in natural chemical processes induced by light. The conical intersection works like a dip switch. For example, if the retina of a human eye is irradiated by light, the electrons start moving, and the molecules of the retinal change their shape, which finally converts the information of light to electrical information for the human brain. The special aspect about conical intersections is that the motion of electrons is transferred to a motion of the atoms very efficiently. And good morning, this is Dr. Will, CBMD, PhD. It's Wednesday, October 26, 2011, 1:27 a.m. Eastern Time. Quick note: the second strong infrared pulse then removed an electron from the molecule, accelerates it, and drives it back to the molecule. As a result, a attosecond light pulse is emitted, which carries a snapshot of the electron distribution in the molecule. And good morning, this is Dr. Will, CBMD, PhD. It's Wednesday, October 26, 2011, 1:28 a.m. Eastern Time. Quick note: the experiment can be compared to photographs which, for example, image a bullet shot through an apple. The bullet would be too fast for the shutter of a camera, resulting in blurred image. Therefore, the shutter is left open, and the picture is illuminated with light flashes, which are faster than the bullet. That's how we get our snapshot. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, October 26, 2011, 5:35 a.m. Eastern Time. Quick note: this defines new globular star clusters and sees right through the heart of the Milky Way. Science Daily, October 20, 2011. 2 newly discovered globular clusters have been added to the total of just 158 known globular clusters in our Milky Way. They were found in new images from ESO Vista survey telescopes as part of the VIA VV survey. This survey was also turned up to the first star cluster that is far beyond the center of the Milky Way, and whose light has had to travel right through the dust and gas in the heart of the galaxy to get to us. And good evening, this is Dr. Will, CBMD, PhD. It's Wednesday, October 26, 2011, 5:38 a.m. Eastern Time. Quick note: as well as globular clusters, VISTA is finding many open or galactic clusters which generally contain fewer younger stars than globular clusters and are far more common. Another newly announced cluster, VVV-CL003, seems to be an open cluster that lies in the direction of the heart of the Milky Way, but much further away, about 15,000 light years beyond the center. This is the first such cluster to be discovered on the far side of the Milky Way. And good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 27, 2011, 2:28 a.m. Eastern Time. Quick note: could a computer one day rewire itself? New nanomaterial steers electrical currents in multiple dimensions. Science Daily, October 16, 2011. Scientists at the Northwestern University have developed a new nanomaterial that can steer electrical currents. The development could lead to a computer that can simply reconfigure its internal wiring and become an entire different device based on changing needs. And good morning, this is Dr. Will, CBMD, PhD. It is Thursday, October 27, 2011, 2:29 a.m. Eastern Time. Quick note: as electronic devices are built smaller and smaller, the materials from which the circuits are constructed begin to lose their properties and begin to be controlled by quantum mechanical phenomena. Reaching the physical barrier, many scientists have begun building circuits into multiple dimensions, such as stacking components on top of one another. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 2:31 a.m. Eastern Time. Quick note: our steering technology allows use to direct current flow through a piece of continuous material. Who led the research like redirecting a river? Streams of electrons can be steered in multiple directions through a block of the material, even multiple streams flowing in opposing directions at the same time. And good morning, this is Dr. Will, CBMD, PhD. It is Thursday, October 27, 2011, 2:33 a.m. Eastern Time. Quick note: imagine a single device that reconfigures itself into a resistor, a rectifier, a diode, and a transistor based on signals from a computer. The multi-dimensional circuitry could be reconfigured into a new electronic circuit using a varied input sequence of electrical pulses. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 2:34 a.m. Eastern Time. Quick note: the hybrid material is composed of electrically conductive particles, each 5 nanomaterials in width, coated with a special positively charged chemical. A nanomaterial is a billionth of a meter. The particles are surrounded by a sea of negatively charged atoms that balance out the positive charges fixed on the particles. By applying an electrical charge across the material, the small negative atoms can be moved and reconfigured, but the relatively larger positive particles are not able to move. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 2:35 a.m. Eastern Time. Quick note: by moving the sea of negative atoms around the material, regions of low and high conductance can be modulated. The result is the creation of a directed path that allows electrons to flow through the material. Old paths can be erased, and new paths created by pushing and pulling the sea of negative atoms. More complex electrical components, such as diodes and transistors, can be made when multiple types of nanoparticles are used. End. Good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 2:47 a.m. Eastern Time. Quick note. It has often been presumed that life developed near the hydrothermal sources known as the black smokers, such those found at the bottom of the oceans along mid-ocean ridges. The abundance of hydrogen, methane, and ammonia produced by these underwater geysers seemed favorable to the emergence of primitive life. Unfortunately, these black smokers are very acid, which prevents amino acid stabilization and thus the formation of organic molecules. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 2:50 a.m. Eastern Time. Quick note. Black smokers are located on ocean ridges. The black appearance of the water stems from the color of the iron and manganese salts that it contains. The Archean eon stretches between 4 and 2.5 billion years ago. Hydration process which enables the formation of serpentinite. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 2:56 a.m. Eastern Time. Quick note. The team recorded data on air temperatures, wind speed, exposed ice and its movement, the emergence of streams, and lakes of meltwater on the surface, and the water's eventual draining away beneath the glacier. This lost meltwater can accelerate the ice sheet slide toward the sea, where it calves new icebergs. Eventually, meltwater reaches the ocean, contributing to the rising sea levels associated with long-term climate change. And good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 27, 2011, 2:57 a.m. Eastern Time. Quick note. On a year that follows one with record-high temperatures, could have more dark ice just below the surface, ready to warm and melt as soon as temperatures begin to rise. This also explains why more ice sheet melting can occur, even though temperatures did not break records. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 3:00 a.m. Eastern Time. Quick note. The team's observations indicate the process was not limited to the glacier they visited. It is a large-scale effect. It is a sign that not only do albedo and other variables play a role in acceleration of melting, but that this acceleration is happening in many places all over Greenland. He cautioned, we are currently trying to understand if this is a trend or will become one. This will help us to improve models projecting future melting scenarios and predict how they might evolve. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 3:09 a.m. Eastern Time. Quick note. Faraway Iris is Pluto's twin. Science Daily, October 26, 2011. Astronomers have accurately measured the diameter of a faraway dwarf planet, for the first time by catching it as it passed in front of a faint star. This event was seen at the end of the 2010 telescopes in Chile. Telescopes at the ESO. The observations show the Iris is an almost perfect twin of Pluto in size. Iris appears to have a very reflective surface, suggesting that it is uniformly covered in a thin layer of ice, probably a frozen atmosphere. The results will be published in the 27th October 2011 issue of the Journal of Nature. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 3:10 a.m. Eastern Time. Quick note. In November 2010, the distant dwarf planet Iris passed in front of a faint background star, an event called inoculation. These occurrences are very rare and difficult to observe, as the dwarf planet is very distant and small. The next such event involving Iris will not happen until 2013. Oculations provide the most accurate and often only way to measure the shape and size of a distant solar system body. And good morning, this is Dr. Will, CBMD, PhD. It says the October 27, 2011, 3:13 a.m. Eastern Time. Quick note. While earlier observations using other methods suggested that Iris was probably about 25% larger than Pluto, with an estimated diameter of 3,000 kilometers, a new study proves that the two objects are essentially the same size. Newly determined diameter strands are 23-26 kilometers with an accuracy of 12 kilometers. This makes its size better than that of its closer counterpart Pluto, which has a diameter estimated to be between 23,000 and 24,000 kilometers. Pluto's diameter is harder to measure because the presence of an atmosphere makes its edge impossible to detect directly by oculations. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 3:16 a.m. Eastern Time. Quick note. The surface of Eris was found to be extremely reflective, resulting 96% of the light that falls on it. A visible albedo of 0.96. This is even brighter than fresh snow on Earth, making Eris one of the most reflective objects in the solar system. along with Saturn's icy moon Enceladus. The bright surface of Eris was most likely composed of a nitrogen-rich ice mixed with frozen methane, as indicated by the object's spectrum, coating the dwarf planet's surface in a thin and very reflective icy layer less than 1 millimeter thick. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 3:17 a.m. Eastern Time. Quick note. This layer of ice could result from the dwarf planet's nitrogen or methane atmosphere condensing as frost onto its surface, as it moves away from the sun in its elongated orbit and into an increasingly cold environment. The icy cold then turned back to a gas. Eris approaches its closest point to the sun at a distance of about 5.7 billion kilometers. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 3:19 a.m. Eastern Time. Quick note. The new results also allow the team to make a new measurement for the surface temperature of a dwarf planet. The estimates suggest a temperature for the surface facing the sun of minus 238 Celsius at most, and an even lower value for the night side of Eris. It is extraordinarily how much we can find out about small and distant objects such as Eris by watching it pass in front of a faint star, using relatively small telescopes, 5 years after the creation of the new class of dwarf planets. We are finally really getting to know one of the founding members. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 3:21 a.m. Eastern Time. Quick note. Notes. 1. TRAPPIST is one of the latest robotic telescopes installed at the El Silla Observatory, with a main mirror just 0.6 meters across. It was inaugurated in June 2010 and is mainly dedicated to the study of exoplanets and comets. The telescope is a project funded by the Belgian Fund for Scientific Research, FNRS. And good morning, this is Dr. Will, CBMD, PhD. Thursday, October 27, 2011, 3:22 a.m. Eastern Time. Quick note. Eris mass is 1.66 times 10 to the power of 22 kilograms, corresponding to 22% of the mass of the moon. For comparison, the moon density is 3.3 grams per centimeter to the power of 3, and water is 1.00 gram per centimeter to the power of 3. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 3:23 a.m. Eastern Time. Quick note. The value of the density suggests that Eris is mainly composed of rock 85% with a small ice content 15%. The latter is likely to be a layer about 100 kilometer thick that surrounds a large rocky core. This very thick layer of mostly water ice is not to be confused with a very thin layer of frozen atmosphere on Eris' surface that makes it so reflective. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 3:24 a.m. Eastern Time. Quick note. The albedo of an object represents the function of light that falls on it and is scattered back into space rather than absorbed. An albedo of 1 corresponds to perfect reflecting white, while 0 is totally absorbing black. For comparison, the moon's albedo is only 0.136, similar to that of coal. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 3:34 a.m. Eastern Time. Quick note. Autistic brains develop more slowly than healthy brains, researchers say. Science Daily, October 20, 2011. Researchers at the UCLA have found a possible explanation for why autistic children act and think differently than their peers. For the first time, they've shown that the connections between brain regions that are important for language and social skills grow much more slowly in boys with autism than in non-autistic children. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 3:38 a.m. Eastern Time. Quick note. Autism is thought to affect 1 in 110 children in the U.S., and many experts believe the numbers are growing. Despite its prevalence, little is known about the disorder, and no cure has been discovered. Normally, as children grow into teenagers, the brain undergoes major changes. This highly dynamic process depends on the creation of new connections called white matter, and the elimination or pruning of unused brain cells called gray matter. As a result, our brains work out the ideal and most efficient ways to understand and respond to the world around us. And good morning, this is Dr. Will, CBMD, PhD. It is Thursday, October 27, 2011, 3:40 a.m. Eastern Time. Quick note. Because the brain of the child with autism develops more slowly during this critical period of life, these children may have an especially difficult time struggling to establish personal identity, develop social interactions, and refine emotional skills. The new knowledge may help to explain some of the symptoms of autism and could improve future treatment options. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 3:41 a.m. Eastern Time. Quick note. The researchers used a type of brain imaging scan called T1 weight MRI, which can map structural changes during brain development, to study how the brains of boys with autism changed over time. They scanned 13 boys diagnosed with autism in a control group of 7 non-autistic boys on 2 separate occasions. The boys ranged in age from 6 to 14 at the time of the first scan. On average, they were scanned again approximately 3 years later. End. Good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 3:43 a.m. Eastern Time. Quick note. By scanning the boys twice, the scientists were able to create a detailed picture of how the brain changes during the critical periods of development. Besides seeing that the white matter connections between these brain regions that are important for language and social skills were growing much slowly. In the boys with autism, they found a second anomaly. In 2 areas of the brain, the putamen, which is involved in learning, and the anterior cingulate, which helps regulate both cognitive and emotional processing, unused cells were not properly pruned away. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 3:48 a.m. Eastern Time. Quick note. Together this creates unusual brain circuits, with cells that are overly connected to their close neighbors and underconnected to important cells further away, making it difficult for the brain to process information in a normal way. The brain's region where growth rates were found to be the most altered were associated with the problems autistic children most often struggle with: social impairment, communication deficits, and repetitive behavior. Future studies using alternative neuroscience techniques should attempt to identify the source of this white matter impairment, the researchers said. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 3:51 a.m. Eastern Time. Quick note. This study provides a new understanding of how the brains of children with autism are growing and developing in a unique way. Brain imaging could be used to determine if treatments are successful at addressing the biological difference. The delayed brain growth in autism may also suggest a different approach for educational intervention in adolescent and adult patients, since we now know their brains are wired differently to perceive information. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 3:59 a.m. Eastern Time. Quick note. Plants feel and force how plants sense touch, gravity, and other physical forces. Science Daily, October 21st, 2011. Picture yourself hiking through the woods or walking across a lawn. Assistant Professor of Biology and Arts and Science at Washington University, St. Louis. Now ask yourself, do the bushes know that someone is brushing past them? Does the grass know that it is being crushed underfoot? Of course, plants don't think thoughts, but they do respond to being touched in a number of ways. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:01 a.m. Eastern Time. Quick note. It is clear that plants can respond to physical stimuli such as gravity or touch. Roots grow down a sensitive plant field, its leaves, and vines twines around the trellis. But we are just beginning to find out how they do it. In the 1980s, work with bacterial cells showed that they have mechanosensitive channels, tiny pores in the cell membranes that open when the cell bloats with water, and the membrane is stretched, letting charged atoms and other molecules rush out of the cell. Water follows the ions, the cell contracts, the membrane relaxes, and the pores close. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:03 a.m. Eastern Time. Quick note. Genes encoding 7 such channels have been found in the bacterium E. coli and 10 Arabidopsis thaliana, small flowering plant related to mustard and cabbage. Both E. coli and Arabidopsis serves as model organism in Haswell's lab. She suspects that there are many more channels yet to be discovered and that they will prove to have a wide variety of functions. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:04 a.m. Eastern Time. Quick note. Swelling bacteria might seem unrelated to folding leaflets, but Haswell is willing to bet they are all related and that mesnosensitive ion channels are at the bottom of them all. After all, plant movements, both fast and slow, are ultimately all hydraulically powered. Where ions go, the water will follow. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:05 a.m. Eastern Time. Quick note. The big problem with studying ion channels has always been their small size, which poses formidable technical challenges. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:05 a.m. Eastern Time. Quick note. Early work in the field done to understand the ion channels, whose coordinated opening and closing creates a nerve impulse, was done in exceptionally large cells, the giant nerve cells of the European squid, which had projections big enough to be seen with the unaided eye. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:06 a.m. Eastern Time. Quick note. Experiments with these channels eventually led to the development of the sensitive electrical recording technique, known as the patch clamp, that allowed researchers to examine the properties of a single ion channel. Patch clamp recording uses as an electrode a glass micropipette that has an open tip. The tip is small enough that it encloses a patch of cell membrane that often contains just one or a few ion channels. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:07 a.m. Eastern Time. Quick note. Patch clamp work showed that there were many different types of ion channels, and that they were involved not just in the transmission of nerve impulses, but also with many other biological processes that involve rapid changes in cells. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:07 a.m. Eastern Time. Quick note. Mechanosensitive channels were discovered when scientists started looking for ion channels in bacteria, which wasn't until the 1980s, because ion channels were associated with nerves, and bacteria weren't thought to have a nervous system. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:09 a.m. Eastern Time. Quick note. In E. coli, the ion channels are embedded in the plasma membrane, which is inside the cell wall. But even if the wall could be stripped away, the cells are far too small to be individually patched, so the work is done with specially prepared giant bacteria cells called spiroplast. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 27, 2011, 4:11 a.m. Eastern Time. Quick note. These are made by culturing E. coli in the broth containing an antibiotic that prevents daughter cells from separating completely within a cell divides. As the cell multiplies, snakes of many cells that share a single plasma membrane form in the culture. If you then digest away the cell wall, they swell up to form a large sphere. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:12 a.m. Eastern Time. Quick note. Not that spiroplasts are big. We are doing most of our studies of Xenopus octopus, frog eggs whose diameters are 150 times bigger than those of spiroplast. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:13 a.m. Eastern Time. Quick note. To find ion channels in bacteria, scientists did electrophysiological surveys of spiroplasts. They stuck a pipette onto a spiroplast and applied suction to the membrane as they looked for tiny currents flowing across the membrane. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:14 a.m. Eastern Time. Quick note. What they found was really amazing. There were 3 different activities that are gated, triggered to open only by deformation of the membrane. They were called activities because nobody knew their molecular or genetic basis. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:14 a.m. Eastern Time. Quick note. The 3 activities were named mechanosensitive channels of large MSCL, small MSCS, and mini MSCM. Conductance, they were distinguished from one another by much tension, had to introduce in order to get them to open and by their conductance. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:16 a.m. Eastern Time. Quick note. Once both genes were known, researchers did knock out experiments to see what happened to the bacteria that didn't have the genes needed to make the channels. What they found was that if both MSCL and MSCS genes were missing, the cells could not survive osmotic downshock, the bacteria equivalent of water torture. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:17 a.m. Eastern Time. Quick note. The standard essay is to grow the bacteria for a couple of generations in very salty broth, so that they have a chance to balance their internal osmolyte concentration with an external one. Osmolytes are molecules that affect osmosis, or the movement of water into and out of the cell. They do this by taking up osmolytes from the environment and making their own. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:21 a.m. Eastern Time. Quick note. You take these bacteria that are chock full of osmolytes and throw them into fresh water. If they don't have the MSCS and MSCL proteins that allow them to jump ions to avoid the uncontrolled influx of water, they don't survive. It is a bit like dumping saltwater fish into a freshwater aquarium. And good morning, it's Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:22 a.m. Eastern Time. Quick note. Why are there 3 mechanosensitive channel activities? The current accepted model is that the channels with the small conductance are the first line of defense. They open early in response to osmotic shock, so that the channels of large conductance through which molecules the call needs can escape. It doesn't open unless it is absolutely necessary. Gradual response thus gives the cell its best chance for survival. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:23 a.m. Eastern Time. Quick note. The next step in the scientific odyssey, figuring out the protein structures, also was very difficult. Protein structures are traditionally discovered by purifying a protein, crystallizing it out of water in solution, and then bombarding the crystal with X-rays. The positions of the atoms and the protein can be deduced from the X-ray diffraction patterns. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:30 a.m. Eastern Time. Quick note. In a sense, crystallizing a protein isn't all that different from growing raw candy from a sugar solution. But as always, the devil is in the details. Protein crystals are much harder to grow than sugar crystals, and once grown, They are extremely fragile. They even can be damaged by an X-ray probes used to examine them. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:34 a.m. Eastern Time. Quick note. And to make things worse, MSCL and MSCS span the plasma membrane, which means that their ends, which are exposed to the paraplasma outside the cell, and the cytoplasm inside the cell, are water-loving, and their middle sections, which are stuck in the greasy membrane, are repelled by water. Because of this double nature, it is impossible to precipitate membrane proteins from water solutions. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:36 a.m. Eastern Time. Quick note. Instead, the technique is to surround the protein with what have been characterized as highly contrived detergents that protect them, but just barely, from the water. Finding the magical balance can take as long as a scientific career. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:36 a.m. Eastern Time. Quick note. The first mechanosensitive channel to be crystallized was the MSCL, not the protein E. coli, but the analogous molecule, a homologue from the bacterium that causes tuberculosis. This work was done in the lab of the Haswell co-authors. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:37 a.m. Eastern Time. Quick note. Of plants and mutants. Up to this point, mechanosensitive channels might not seem all that interesting because the lives of bacteria are not of supreme interest to us unless they are making us ill. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:39 a.m. Eastern Time. Quick note. Scientists began to compare the genes for the bacteria channels to the genomes of other organisms, and they discovered that there are homologous sequences not just in other bacteria but also in some multicellular organisms, including plants. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:39 a.m. Eastern Time. Quick note. I was interested in gravity and touch responses in plants. I saw these papers and thought these homologues were great candidates for proteins that might mediate those responses. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:40 a.m. Eastern Time. Quick note. There are 10 MSCS homologues in the Arabidopsis and no MSCL homologues. What's more, different homologues are found not just in the cell membrane, but also in chloroplast and mitochondrial membranes. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:42 a.m. Eastern Time. Quick note. The chloroplast is a light-capturing organelle, and the plant cell and the mitochondria is its power station. Both are thought to be balanced independent organisms that were engulfed and enslaved by cells which found them useful. Their membranes are vestiges of their free-living past. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 27, 2011, 4:42 a.m. Eastern Time. Quick note. The number of homologues and their locations in plant cells suggests these channels do much more than prevent the cells from taking on board too much water. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:44 a.m. Eastern Time. Quick note. From these mutants, she learned that 2 of the 10 channels control chloroplast size and proper division, as well as a leaf shape. Plants with mutations in these 2 MSCS channel homologues have giant chloroplasts that haven't divided properly. The monster chloroplast garnered her lab the cover of the August issue of the Plant Cell. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:45 a.m. Eastern Time. Quick note. We showed that bacteria lacking MSCS and MSCL don't divide properly either. So the link between these channels and division is evolutionary conserved. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:46 a.m. Eastern Time. Quick note. The big idea. Authors think they are only scratching the surface. We are basing our understanding of the class of channels of the MSCS itself, which is a very reduced form of the channel. It is relatively tiny. And good morning, it's Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:47 a.m. Eastern Time. Quick note. But we know that some of the members of this family have long extensions that stick out from the membrane, either outside or inside of the cell. We suspect this means that the channels not only discharge ions, but that they also signal to the whole cell in other ways. They may be integrated into common signaling pathways, such as the cellular osmotic stress response pathway. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:51 a.m. Eastern Time. Quick note. We think we may be missing a lot of complexity by focusing too exclusively on the first members of this family of proteins to be found and characterized. We think there's a common channel core that makes these proteins respond to membrane tension, but that all kinds of functionality relevant regulation may be layered on top of that. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 4:52 a.m. Eastern Time. Quick note. There is a channel in E. coli that's closely related to MSCS that has huge extension outside the cell that makes it sensitive to potassium. So it is a mechanosensitive channel, but it only gates in the presence of potassium. What that's important for, we don't know yet, but it tells us there are other functions out there we haven't studied. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 5:16 a.m. Eastern Time. Quick note. Face-to-face with an ancient human. Science Daily, October 20, 2011. A reconstruction based on the skull of Norway's best preserved Stone Age skeleton makes it possible to study the features of a boy who lived outside Stavanger 7 500 years ago. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 5:19 a.m. Eastern Time. Quick note. 3D simulations of nova explosions. Science Daily, October 20, 2011. A new study has shown how mixing of elements occurs during a nova explosion, thus solving an enigma that has puzzled cellular astrophysicists for over half a century. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 27, 2011, 5:24 a.m. Eastern Time. Quick note. As a result of the complex nuclear phenomena that take place inside stars, the universe has evolved from a chemically poor state, dominated exclusively by the presence of hydrogen, helium, and traces of lithium, to one containing nearly 100 stable elements, without the variety of elements now present in the cosmos, the formation of structures like planets and stars, and the genesis of life forms, including the calcium in our bones, the iron in our blood, and the uranium we use in nuclear power plants, would have been impossible. Most of chemical elements originated in supernova and nova titanic stellar explosions. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 5:26 a.m. Eastern Time. Quick note. Nova and cataclysmic stellar phenomena that take place in binary systems consisting of a compact stellar object, a white dwarf star the size of a planet but with a mass of up to 1.4 times that of the Sun, and a low-mass star. The stars must be close enough for the intense gravitational field of the white dwarf to tear material away from the outer layers of its companion. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 5:27 a.m. Eastern Time. Quick note. Nova, which are relatively frequent in our galaxy, some 30 to 35 nova-like explosions occur each year, are the third most energetic stellar explosions in the universe, after supernova and gamma-ray burst. Nova and supernova have been observed by humans for over 2,000 years. The way these stars suddenly become much brighter, a change sometimes observable to the naked eye, has given rise to a great variety of conjectures about their origin. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 5:33 a.m. Eastern Time. Quick note. With the advent of new, more precise observational techniques such as photometry and spectroscopy, experts have been able to precisely characterize some physical properties of the material ejected during nova explosions, such as its chemical composition. Scientists know that the material transferred by the companion star is often a solar composition, close to 98% hydrogen and helium by mass, but other elements in the range between carbon C and calcium Ca on the periodic table can account for 30% or to 50% of the material ejected during a nova explosion. And good morning, this is Dr. Will, CBMD, PhD, Thursday, October 27, 2011, 5:39 a.m. Eastern Time. Quick note. The origin of this peculiar pattern of chemical abundances and their irregular distribution of nova ejecta is an enigma in the field of stellar astrophysics that has eluded explanation for almost half a century. According to the most likely hypothesis, when the material transferred by the companion star piles up on the white dwarf, mixing episodes occur at the interface between the outer layers of the white dwarf and the envelope of transferred material. But the characterization of the mixing process has provoked vivid discussions. One of the proposed mechanisms could not be rigorously simulated until sufficiently powered computational tools were available. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 27, 2011, 5:46 a.m. Eastern Time. Quick note. The accumulation of material on the white dwarf VIA, this mechanism is unstable. This instability results in mixing episodes involving material at the interface between the outer layers of the white dwarf, rich in elements like carbon and oxygen, or oxygen and neon, and the envelope is transferred material. The researchers were also able to establish the extent of heavy element enhancement caused by this phenomenon, which up to now had been deduced from the observational data. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 27, 2011, 5:50 a.m. Eastern Time. Quick note. This phenomenon was demonstrated based on 3D simulations of the mixing process, which the team performed for the first time for supernova explosions. The simulations were possible thanks to the use of sophisticated tools like the Mare Nostrum computer at the Barcelona Supercomputing Center, Central National Supercomputation, BSC, CNS, after 150,000 hours of calculation. And Good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 5:52 a.m. Eastern Time. Quick note. The possibility of recreating 3D physical phenomenon like convection under the conditions that prevail during nova explosions also led to a solution to the second part of the puzzle. Scientists already knew how energy transport by convection works in stars, but could offer only a formal account of this phenomenon. Now, thanks to the study conducted by UPC researchers, it has been numerically shown that under the conditions that exist during a nova explosion, the matter plasma is in a turbulent state. Its movement is unstructured, almost chaotic. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, October 27, 2011, 5:58 a.m. Eastern Time. Quick note. The intermittent nature of this turbulence results in irregularities in the chemical distribution of material in injected envelopes. Up until now, this phenomenon had never been numerically tested in nova explosions. In fact, it had not even been proposed as a hypothesis to explain the origin of the observed chemical heterogeneity. The heterogeneity was attributed until now to lack of precision in the measuring process. The UPS study shows that this phenomenon is real, and that it is caused by the intermittent nature of the turbulent phenomenon that occur in stellar plasma as it undergoes a thermonuclear explosion. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 3:59 a.m. Eastern Time. Quick note. Nanofunnels used to generate extreme ultraviolet light pulses. Science Daily, October 17, 2011. If you want to avoid spilling when you are pouring liquids in the kitchen, you may appreciate a funnel. Funnels are not only useful tools in the kitchen, light can also be efficiently concentrated with funnels. In this case, the funnels have to be about 10,000 times smaller. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 4:01 a.m. Eastern Time. Quick note. To concentrate the energy of infrared light pulses with a nanofunnel and use the concentrated energy to generate extreme ultraviolet light flashes. These flashes, which repeated 75 million times per second, lasted only a few femtoseconds. The new technology can help in the future to measure the movement of electrons with the highest spatial and temporal resolution. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 4:02 a.m. Eastern Time. Quick note. Light is convertible. The wavelengths composing the light can change through interactions with matter, where both the type of material and shape of material are important for frequency conversion. An international team of scientists from Korea advances to the science technology. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 4:03 a.m. Eastern Time. Quick note. The scientists converted femtosecond laser pulses in the infrared spectral range to femtosecond light flashes in the extreme ultraviolet EUV. Ultra-short pulsed EUV light is used in laser physics to explore the inside of atoms and molecules. A femtosecond lasts only a millionth of a billionth of a second. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 4:04 a.m. Eastern Time. Quick note. Light in the infrared IR can be converted to the EUV by a process known as high harmonic generation, whereby the atoms are exposed to a strong electric field from the IR laser pulses. Those fields have to be as strong as the fields holding the atom together. With these fields, electrons can be extracted from the atoms and accelerated with full force back onto the atoms. Upon impact, highly energetic radiation and the EUV is generated. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 4:05 a.m. Eastern Time. Quick note. To reach the necessary strong electric fields for the production of EUV light, the team scientist has now combined the scheme with a nanofunnel in order to concentrate the electric field of the light. With their new technology, they were able to create a powerful EUV light source with wavelengths down to 20 nanometers. The light source exhibits a so far unreached high repetition rate. The few femtoseconds lasting EUV light flashes are repeated 75 million times per second. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 4:07 a.m. Eastern Time. Quick note. The core of the experiment was a small, only few micrometers long, slightly elliptical funnel made out of silver and filled with xenon gas. The tip of the funnel was only Ca, 100 nanometers wide. The infrared light pulses were sent into the funnel entrance, where they travel through towards the small exit. The electromagnetic forces of light result in density fluctuations of the electrons on the inside of the funnel. Here, a small patch of the metal surface was positively charged, the next one negative, and so on, resulting in a new electromagnetic fields on the inside of the funnel, which are called surface plasmon polaritons. The surface plasmon polaritons travel toward the tip of the funnel, where the conical shape of the funnel results in the concentration of their fields. The field on the inside of the funnel can become a few hundred times stronger than the field of the incident infrared light. This enhanced field results in the generation of EUV light and the XE gas end. Good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011 at 4:09 a.m. Eastern Time. Quick note. The nano funnel has yet another function. Its small opening at the exit acts as a doorman for light wavelengths. Not every opening is passable for light. If the opening is smaller than half of the wavelength, the other side remains dark. The 100 nanometer large opening of the funnel did not allow the infrared light at 800 nanometers to pass. The generated EUV pulses with wavelengths down to 20 nanometers passed. However, without problems, the funnel acts as an efficient wavelength filter. At the small opening, only EUV light comes out. End. Good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 4:21 a.m. Eastern Time. Quick note. Due to their short wavelengths and potentially short pulses, duration reaching into the attosecond domain, extreme ultraviolet light pulses are an important tool for the exploration of electron dynamics in atoms, molecules, and solids. Explains. Electrons are extremely fast moving on attosecond timescales, attosecond and a billionth of a second. In order to capture a moving electron, light flashes are needed, which are shorter than the time scale of the motion. Attosecond light flashes have become a familiar tool in the exploration of electron motion. With the conventional techniques, they can only be repeated a few thousand times per second. This can change with the nano funnel. We assume that the few femtosecond light flashes consist of trains of attosecond pulses. End. Good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 4:25 a.m. Eastern Time. Quick note. The repetition rate is important for e.g. application of EUV pulses in electron spectroscopy on surfaces. Electrons repel each other by Coulomb forces. Therefore, it may be necessary to restrict the experimental conditions such that only a single electron is generated per laser shot. With low repetition rates, long data acquisition times would be required in order to achieve sufficient experimental resolution. In order to conduct experiments with high spatial and temporal resolution within a sufficiently short time, a high repetition rate EUV source is needed. The novel combination of laser technology and nanotechnology can help in the future to record movie of ultra-fast electron motion on surfaces with so far unreached temporal and spatial resolution in the attosecond nanometer domain. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 4:33 a.m. Eastern Time. Quick note. How the Milky Way killed off nearby galaxies. Science Daily, October 18, 2011. Two researchers from Observatoria Strasbourg have revealed for the first time the existence of a new signature of the birth of the first stars in our galaxy, the Milky Way, more than 12 billion years ago. The intense ultraviolet light from these stars dispersed the gas of our galaxy's nearest companions, virtually putting a halt to their ability to form stars and consigning them to a dim future. And good morning, this is Dr. Will, CBMD, PhD. It's Friday, October 21st, 2011, 4:36 a.m. Eastern Time. Quick note. The first stars of the universe appeared about 150 million years after the Big Bang. Back then, the hydrogen and helium gas filling the universe was cold enough for its atoms to be electrically neutral. As the ultraviolet UV light of the first stars propagated through this gas, it broke apart the proton-electron pairs that make up hydrogen atoms, returning them to the so-called plasma state they experienced in the first moments of the universe. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 4:37 a.m. Eastern Time. Quick note. This process, known as reionization, also resulted in a significant heating which had dramatic consequences. The gas became so hot that it escaped the weak gravity of the lowest mass galaxies, thereby depriving them of the material needed to form stars. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 4:38 a.m. Eastern Time. Quick note. It is now widely accepted that this process can explain the small number and large ages of the stars seen in the faintest dwarf galaxy satellites of the Milky Way. It also helps scientists understand why galaxies like the Milky Way have so few satellites around them. The missing satellites problem, the stripping out of gas from these galaxies, makes them sensitive probes of the UV radiation and the reionization approach. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 4:40 a.m. Eastern Time. Quick note. The satellite galaxies are also relatively close, from 30,000 to 900 light years away, which allows us to study them in great detail. something that will be enhanced by the coming generation of larger telescopes. Comparing the population of their stars and each galaxy with its position could give us a unique insight into the structure of the UV radiation emitted from the earliest stars in the Milky Way. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 4:41 a.m. Eastern Time. Quick note. Until now, models for this process assume that the radiation leading to the removal of gas from galaxy satellites was produced collectively by all the large galaxies, nearby resulting in a uniform background of UV light. The new model put together by the two French researchers proves this assumption wrong. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 4:43 a.m. Eastern Time. Quick note. Invisible dark matter that makes up about 23% of the universe structured itself with the stars in our galaxy and its environs. From shortly after the Big Bang to the present day, they used the high-resolution numerical simulation to model the formation of stars and gas trapped in dark matter halos that envelope galaxies, and then to describe how this gas reacted to UV radiation. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 4:44 a.m. Eastern Time. Quick note. This is the first time that a model accounts for the effect of the radiation emitted by the first stars formed at the center of the Milky Way on its satellite galaxies. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 4:44 a.m. Eastern Time. Quick note. In contrast to previous models, the radiation field produced is not uniform, but decreases in its intensity as one moves away from the center of the Milky Way. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 4:46 a.m. Eastern Time. Quick note. The satellite galaxy close to the galactic center see their gas evaporate very quickly. They form so few stars that they can be undetectable with current telescopes. At the same time, the more remote satellite galaxies experience, on average, a weaker irradiation. Therefore, they manage to keep their gas longer and form more stars. As a consequence, they are easier to detect and appear more numerous. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 4:47 a.m. Eastern Time. Quick note. The new model appears to be a close match to observations of our galaxy and its neighborhood, and suggests that the first stars of our galaxy played a major role in the photoevaporation of the satellite galaxies. It is not large nearby galaxies, but our own, that caused the demise of its tiny neighbors, as fixating them through its intense radiation. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 5:18 a.m. Eastern Time. Quick note. Computing building blocks created from bacteria and DNA. Science Daily, October 18, 2011. Scientists have successfully demonstrated that they can build some of the basic components for digital devices out of bacteria and DNA, which could pave the way for a new generation of biological computing devices and research published October 18th in the Journal of Nature Communications. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 5:18 a.m. Eastern Time. Quick note. The researchers from the Imperial College of London have demonstrated that they can build logic gates, which are used for processing information in devices such as computers and microprocessors, out of harmless gut bacteria and DNA. These are the most advanced biological logic gates ever created by scientists. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 5:20 a.m. Eastern Time. Quick note. Logic gates are the fundamental building blocks in silicon circuitry that our entire digital age is based on. Without them, we could not process digital information. Now that we have demonstrated that we can replicate these parts using bacteria and DNA, we hope that our work could lead to a new generation of biological processors, whose applications in information processing could be as important as their electronic equivalents. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 5:22 a.m. Eastern Time. Quick note. Although still a long way off, the team suggests that these biological logic gates could one day form the building blocks in microscopic biological computers. Devices may include sensors that swim inside arteries, detecting the buildup of harmful plaque and rapidly delivering medications to the affected zone. Other applications may include sensors that detect and destroy cancer cells inside the body, and pollution monitors that can be deployed in the environment, detecting and neutralizing dangerous toxins such as arsenic. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 5:23 a.m. Eastern Time. Quick note. Previous research only proved that biological logic gates could be made. The team say that the advantage of their biological logic gates over previous attempts is that they behave more like their electronic counterparts. The new biological gates are also modular, which means that they can be fitted together to make different types of logic gates, paving the way for more complex biological processors to be built in the future. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 5:26 a.m. Eastern Time. Quick note. In a new study, the researchers demonstrated how these biological logic gates work. In one experiment, they showed how biological logic gates can replicate the way that electronic logic gates process information by either switching on or off. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 5:28 a.m. Eastern Time. Quick note. The scientists constructed a type of logic gate called an AND gate from bacteria called Escherichia coli, E. coli, which is normally found in the lower intestine. The team altered the E. coli with modified DNA, which reprogrammed it to perform the same switching on and off process as its electronic equivalent when stimulated by chemicals. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 5:29 a.m. Eastern Time. Quick note. The researchers were also able to demonstrate that the biological logic gates could be connected together to form more complex components in a similar way that electronic components are made. In another experiment, the researchers created a NOT gate and combined it with the AND gate to produce the more complex NAND gate. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 5:30 a.m. Eastern Time. Quick note. The next stage of the research will see the team trying to develop more complex circuitry that comprises multiple logic gates. One of the challenges faced by the team is finding a way to link multiple biological logic gates together, similar to the way in which electronic logic gates are linked together, to enable complex processing to be carried out. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 5:38 a.m. Eastern Time. Quick note. NASA Spicer detects comet storm in nearby solar system. Science Daily, October 19, 2011. NASA Spicer Space Telescope has detected signs of icy bodies raining down in an alien solar system. The downpour resembles our own solar system several billion years ago during a period known as the Late Heavy Bombardment, which may have brought water and other life-forming ingredients to Earth. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 5:40 a.m. Eastern Time. Quick note. During this approach, comets and other frosty objects that were flung from the outer solar system pummeled the inner planets. The barrage scared our moon and produced large amounts of dust. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 5:41 a.m. Eastern Time. Quick note. Astronomers use Spicer infrared detectors to analyze light coming from the dust around the ETA. Certain chemical fingerprints were observed, including water, ice, organics, and a rock, which indicate a giant comet source. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 5:41 a.m. Eastern Time. Quick note. The light signature emitted by the dust around the ETA also resembles the meteorite, which fell to Earth in fragments across Sudan in 2008. The similarities between the meteorite and the object obliterated, the ETA, imply a common birthplace in their respective solar systems. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 5:43 a.m. Eastern Time. Quick note. A second, more massive ring of colder dust located at the far edge of the ETA system seems like a proper environment for a reservoir of cometary bodies. This bright ring, discovered in 2005, looms at about 150 times the distance from ETA. Earth is from the Sun. Our solar system has a similar region, known as the Belt, where the icy, rocky leftovers from planet formation linger. The new Spicer data suggests that the meteorite may have originated in our own belt. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 5:45 a.m. Eastern Time. Quick note. The belt was home to a vastly greater number of these frozen bodies, collectively dubbed belt objects, about 4 billion years ago, some 600 million years after our solar system formed. Scientists think the belt was disturbed by a migration of the gas giant planets Jupiter and Saturn. This jarring shift in the solar system's gravitational balance scattered the icy bodies in the Kepler belt, fling the vast majority into the interstellar space and producing cold dust in the belt. Some Kepler belt's objects, however, were set on paths that crossed the orbits of the inner planets. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 5:48 a.m. Eastern Time. Quick note. The resulting bombardment of comets lasted until 3.8 billion years ago. After comets impacted the side of the moon that faces Earth, magma seeped out of the lunar crust, eventually cooling into dark seas, or Maria. When viewed against the lighter surrounding areas of the lunar surface, those seas form the distinctive men in the moon visage. Comets also struck Earth or incinerated in the atmosphere and are thought to have deposited water and carbon on our planet. This period of impacts might have helped life form by delivering its crucial ingredients. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 5:49 a.m. Eastern Time. Quick note. We think the ETA system should be studied in detail to learn more about the rain of impacting comets and other objects that may have started life on our own planet. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 6:18 a.m. Eastern Time. Quick note. U.S. rivers and streams saturated with carbon. Science Daily, October 19, 2011. Rivers and streams in the United States are releasing substantially more carbon dioxide into the atmosphere than previously thought, according to researchers publishing their results in the current issue of the Journal of Nature Geoscience. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 6:19 a.m. Eastern Time. Quick note. Their findings could change the way scientists model the movement of carbon among land, water, and the atmosphere. Direct measurements of carbon dioxide concentrations and fluxes in streams and rivers are still extremely rare. Program Director, National Science Foundation, NSF, Division of Environmental Biology, which funded the research. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 6:20 a.m. Eastern Time. Quick note. This study demonstrates that both are much higher than assumed. The research should enable more predictive and precise models of carbon cycling at regional to global scales. The researchers found that a significant amount of carbon contained in land, which first is absorbed by plants and forests through the air, is leaking into streams and rivers, and then released into the atmosphere before reaching coastal waterways. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 6:22 a.m. Eastern Time. Quick note. What we are able to show is that there is a source of atmospheric carbon dioxide from streams and rivers, and that it is significant enough for terrestrial modelers to take note of it. He and his co-author, ecologist Peter Raymond, also at Yale analyzed data from samples of more than 4,000 rivers and streams throughout the United States and incorporated detailed geospatial data to model the flux of carbon dioxide from the water. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 6:24 a.m. Eastern Time. Quick note. This release is equal to a car burning 40 billion gallons of gasoline, enough to drive back and forth to the moon 3.4 million times. These rivers breathe a lot of carbon. They are a source of carbon dioxide just like we breathe out carbon dioxide, like smokestacks emit carbon dioxide. This has never been systematically estimated from a region as large as the United States. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 6:25 a.m. Eastern Time. Quick note. The paper titled Significant Efflux of Carbon Dioxide from Streams and Rivers in the United States also indicates that as the climate heats up, there will be more rain and snow, and that an increase in precipitation will result in even more terrestrial carbon flowing into rivers and streams and being released into the atmosphere. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 6:26 a.m. Eastern Time. Quick note. Any accurate estimate of carbon uptake versus carbon released must include the carbon in streams and rivers. The researchers note that currently it is difficult to determine how to include this flux in regional carbon budgets because the influence of human activity on the release of carbon dioxide into streams and rivers is still unknown. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 7:36 a.m. Eastern Time. Quick note. Young human-specific genes correlated with brain evolution. Science Daily, October 19, 2011. Young genes that appeared since the primate branch split from other mammal species are expressed in unique structures of the developing human brain, a new analyst finds. The correlation suggests that scientists studying the evolution of the human brain should look to genes considered recently by evolutionary standards in early stages of brain development. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 7:38 a.m. Eastern Time. Quick note. Scientists have long sought to solve how the brain evolved to have the anatomical features and functional ability that separate humans from their primate ancestors. With the completion of Human Genome Project in 2003 and the growing availability of genome sequences for primates and other species, researchers have looked to genetics for answers on brain evolution. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 7:40 a.m. Eastern Time. Quick note. From these studies, many scientists have hypothesized that differential regulation of conserved genes shared across species, rather than the arrival of new species-specific protein-encoding genes, was responsible for dramatically different human brain. But in the 2010 study, Long Laboratory discovered that the younger species-specific gene could be just as important as older conserved genes to an organism's development. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 7:41 a.m. Eastern Time. Quick note. For the PLOS Biology paper, researchers merged a database of gene age with transcription data from humans and mice to look for when and where young genes specific to each species were expressed. The researchers found that a higher percentage of primate-specific young genes were expressed in the brain compared to mouse-specific young genes. Human-specific young genes also were more likely to be expressed in the recently expanded human brain structures, such as the neocortex and prefrontal cortex. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 7:44 a.m. Eastern Time. Quick note. The timing of when the young human-specific genes are expressed in the brain also intrigued the researchers, inspired by an ultrasound appointment with his pregnant wife, calculated when young genes were expressed in the human brain, discovering that they were more likely to appear during fetal or infant development. The early activity of these genes suggests scientists should be looking at earlier developmental stages for genetic activity that ultimately shapes the complexity of the human brain. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 7:45 a.m. Eastern Time. Quick note. What's really surprising is that the evolutionary newest genes on the block act early. Graduate students in Long Laboratories, primate-specific genes act before birth, even when a human embryo doesn't look very different from a mouse embryo, but the actual differences are laid out early. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 7:46 a.m. Eastern Time. Quick note. Thus far, researchers comparing adult brains between species have focused on regulatory differences as primary driver of evolutionary changes. But the new research suggests that new genes with novel functions may have also played an important, previously overlooked role in the evolution of the human brain. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 7:47 a.m. Eastern Time. Quick note. Traditionally, people don't believe that a new protein or a new gene can play any role in an important process. Most people pay attention to only the regulation of genes. But out of a total of about 1,300 new genes, only 13% were involved in new regulation. The rest, some 1,100 genes, are new genes that bring a whole new type of function. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 7:48 a.m. Eastern Time. Quick note. Future research will look at the function of these genes and the role they play in building the unique human brain. People tend to study genes that are old functions present in organisms. This work will open up a new window such that people will start working in these new genes to try to figure out what exactly the functions are. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 7:49 a.m. Eastern Time. Quick note. For now, the authors stress that their finding is only a correlation between the appearance of young human-specific genes and the evolutionary appearance of advanced brain structures. More data will need to be collected on the timing and location of gene expression in non-human primates to determine precisely which new genes and biological functions contributed to the evolution of the human brain. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 7:50 a.m. Eastern Time. Quick note. We don't know if this observation has any causation biologically, and there is a long way to go from there. But this correlation can predict some future work to do. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 9:39 a.m. Eastern Time. Quick note. Bioluminescence explanation for glowing sea suggested. Science Daily, October 19, 2011. It has long been known that distinctive blue flashes, a type of bioluminescence that are visible at night in some marine environments, are caused by tiny unicellular plankton known as dinoflagellates. However, a new study has for the first time detailed the potential mechanism for this bioluminescence. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 9:40 a.m. Eastern Time. Quick note. A key aspect of the potential mechanism for bioluminescence and dinoflagellates proposed in the PNAS study involves voltage-graded proton channels in membranes that can be opened or closed by chemical or electrical events. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 9:42 a.m. Eastern Time. Quick note. According to the study, here is how the light-generating process in dinoflagellates may work. As dinoflagellates float, mechanical stimulation generated by the movement of surrounding water sends electrical impulses around an internal compartment within the organism called a vacuole, which holds an abundance of protons. These electrical impulses open so-called voltage-sensitive proton channels that connect the vacuole to tiny pockets dotting the vacuole membrane, known as scintillons. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 9:43 a.m. Eastern Time. Quick note. Once open, the voltage-sensitive proton channels may funnel protons from the vacuole into the scintillons. Protons entering the scintillons then activate luciferase, a protein which produces flashes of light. That is stored in scintillons. Flashes of light produced by resulting luciferase activation would be most visible during blooms of dinoflagellates. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 9:44 a.m. Eastern Time. Quick note. This research illuminates the novel mechanism underlying a beautiful natural phenomenon in our oceans and enhances our understanding of dinoflagellates, some of which can produce toxins that are harmful to the environment. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 9:47 a.m. Eastern Time. Quick note. Luhmann classifies this object as a brown dwarf, an object that formed just like a star out of a massive cloud of dust and gas. But the mass that a brown dwarf accumulates is not enough to ignite thermonuclear reactions in its core, resulting in a failed star that is very cool. In the case of the new brown dwarf, the scientists have gauged the temperature of its surface to be between 80 and 160 degrees Fahrenheit, possibly as cool as a human. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 9:48 a.m. Eastern Time. Quick note. Ever since brown dwarfs first were discovered in 1995, astronomers have been trying to find new record holders for the coldest brown dwarfs, because these objects are valuable as laboratories for studying the atmospheres of planets with Earth-like temperatures outside our solar system. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 9:49 a.m. Eastern Time. Quick note. Astronomers have named the brown dwarf WD 0806661b because it is the orbiting companion of an object named WD 0806661, the white dwarf core of the star that was like the Sun until its outer layers were expelled into space during the final phase of its evolution. The distance of this white dwarf from the Sun is 63 light years, which is very near our solar system, compared with most stars in our galaxy. And good morning, this is Dr. Will, CBMD, PhD. It's Friday, October 21st, 2011, 9:50 a.m. Eastern Time. Quick note. The distance of this white dwarf from its brown dwarf companion is 2,500 astronomical units, AU, about 2,500 times the distance between the Earth and the Sun. So its orbit is very large as compared with the orbits of planets, which form within a disk of dust swirling close around the newborn star. University of California, San Diego, a member of the Discovery team, because it has such a large orbit. The astronomers say this companion most probably was born in the same manner as binary stars, which are known to be separated as far apart as this pair, while remaining gravitationally bound to each other. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 9:51 a.m. Eastern Time. Quick note. Luhmann and his team colleagues presented this new candidate for the coldest known brown dwarf in a paper published in spring 2011, and they now have confirmed its record-setting cool temperature in a paper that will be published in the Astrophysical Journal. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 9:53 a.m. Eastern Time. Quick note. To make their discovery, Luhmann and his colleagues searched through infrared images of over 600 stars near our solar system. They compared images of nearby stars taken a few years apart, searching for any faint points of light that showed the same motion across the sky as the targeted star. Objects with cool temperatures like the Earth are brightest at infrared wavelengths. We use NASA's Spicer Space Telescope because it is the most sensitive infrared telescope available. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 9:54 a.m. Eastern Time. Quick note. In a related new discovery involving a different cool brown dwarf, his colleagues have made the most detailed measurement yet of ammonia in the atmosphere of an object outside our solar system. These new data are much higher quality than previously achieved, making it possible to study in much more detail than ever before the atmospheres of the coldest brown dwarfs, which most closely resemble the atmospheres that are possible around planets. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 9:56 a.m. Eastern Time. Quick note. Brown dwarfs that are far from their companion stars are much easier to study than our planets, which typically are difficult to observe because they get lost in the glare of the stars they orbit. Brown dwarfs with Earth-like temperatures allow us to refine theories about the atmospheres of objects outside our solar system that have comparatively cool atmospheres like that of our own planet. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 21st, 2011, 10:40 a.m. Eastern Time. Quick note: nearby planet-forming disk holds water for thousands of oceans. Science Daily, October 20, 2011. For the first time, astronomers have detected around a burgeoning solar system spalling cloud of water vapor that's cold enough to form comets, which could eventually deliver oceans to dry planets. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 10:41 a.m. Eastern Time. Quick note: water is an essential ingredient for life. Scientists have found thousands of Earth oceans worth of it within the planet-forming disk surrounding the star TW. TW Hydra is 176 light-years away in the constellation Hydra, and is the closest solar system to be. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 10:42 a.m. Eastern Time. Quick note: the researchers used the heterodyne instrument for the far-infrared HiFi on the orbiting Herschel Space Observatory to detect the chemical signature of water. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 10:44 a.m. Eastern Time. Quick note: scientists had previously found warm water vapor in planet-forming disks close to the central star, but until now evidence for vast quantities of water extending into the cooler, far reaches of disk where comets and giant planets take shape had not emerged. The more water available in disk for icy comets to form, the greater the chances that large amounts will eventually reach new planets through impacts. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 10:45 a.m. Eastern Time. Quick note: the detection of water sticking to dust grains throughout the planet-forming disk would be similar to events in our own solar system's evolution, where over millions of years, these dust grains would then coalesce to form comets. These would be a prime delivery mechanism for water on planetary bodies. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 10:45 a.m. Eastern Time. Quick note: other recent findings from HiFi support the theory that comets delivered a significant portion of Earth's oceans. Researchers found that the ice on the comet called Hartley 2 has the same chemical composition as our oceans. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 10:46 a.m. Eastern Time. Quick note: HiFi is helping astronomers gain a better understanding of how water comes to terrestrial planets, Earth and beyond, if TW Hydra and its icy disk are representative of many other young star systems. As researchers think they are, then the process for creating planets around numerous stars with abundant water throughout the universe appears to be in place, NASA officials say. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 21st, 2011, 3:10 p.m. Eastern Time. Quick note: significant ozone hole remains over Antarctica. Science Daily, October 20th, 2011. The Antarctic ozone hole, which yawns wide every southern hemisphere spring, reached its annual peak on September 12th, stretching 10.05 million square miles, the ninth largest on record above the South Pole. The ozone hole reached its steepest point of the season on October 9, 2011, when total ozone readings dropped to 102 Dobson units, tying for the 10th lowest in the 26-year record. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 3:12 p.m. Eastern Time. Quick note: the ozone layer helps protect the planet's surface from harmful ultraviolet radiation. NOAA and the NASA used balloon-borne instruments, ground instruments, and satellites to monitor the annual South Pole ozone hole, global levels of ozone in the stratosphere, and the human-made chemicals that contribute to ozone depletion. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 3:12 p.m. Eastern Time. Quick note: the upper part of the atmosphere over the South Pole was colder than average this season, and that cold air is one of the key ingredients for ozone destruction. Monitoring Division in Boulder, Colorado. Other key ingredients are ozone-depleting chemicals that remain in the atmosphere and ice crystals on which ozone-depleting chemical reactions take place. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 3:13 p.m. Eastern Time. Quick note: even though it was relatively large, the size of the year's ozone hole was within range we'd expect given the levels of man-made ozone-depleting chemicals that continue to persist. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 3:15 p.m. Eastern Time. Quick note: In August and September, spring Antarctica, the sun begins rising again after several months of darkness. Circumpolar winds keep cold air trapped above the continent. Sunlight sparkled reaction involving ice clouds. Human-made chemicals begin eating away at the ozone. Most years, the conditions for ozone depletion ease by early December and seasonal hole at closeness. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 3:16 p.m. Eastern Time. Quick note. Levels of most ozone-depleting chemicals in the atmosphere have been gradually declining since an international treaty to protect the ozone layer. The 1987 Montreal Protocol was signed. The international treaty caused the phase-out of ozone-depleting chemicals, then used widely in refrigeration as solvents and in aerosol spray cans. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 3:18 p.m. Eastern Time. Quick note. Global atmospheric models predict that stratospheric ozone could recover by the middle of the century, but the ozone hole in the Antarctica will likely persist 1 to 2 decades beyond that, according to the latest analysis by the World Meteorological Organization 2010 ozone assessment, with the co-authors from NOAA and NASA. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 3:19 p.m. Eastern Time. Quick note. Researchers do not expect a smooth, steady recovery of Antarctica ozone because of natural ups and downs in temperatures and other factors that affect depletion, noted NOAA-ESRL scientist Brian Johnson. Johnson helped co-author a recent NOAA paper that concluded it would take another decade to begin discerning changes in the rates of ozone depletion. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 3:24 p.m. Eastern Time. Quick note. Astronomers explain blue staggerers. How do mysterious stars stay so young? Science Daily, October 20, 2011. Mysterious blue staggerers are old stars that appear younger than they should be. They burn hot and blue. Several theories have attempted to explain why they don't show their age, but until now scientists have lacked the crucial observations with which to test each hypothesis. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 3:25 p.m. Eastern Time. Quick note. Armed with such observational data, 2 astronomers from Northwestern University and University of Wisconsin-Madison report that a mechanism known as mass transfer explains the origins of blue staggerers. Essentially, a blue staggerer eats up the mass, or outer envelope, of its giant star companion. This extra fuel allows staggerer to continue to burn and live longer while the companion star is stripped bare, leaving only its white dwarf core. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 3:27 p.m. Eastern Time. Quick note. The majority of blue staggerers in their study are in binaries. They have a companion star. It is really the companion star that helped us determine where the blue staggerers comes from. Northwestern astronomer first authored the study. The companion stars orbit as periods of about 1,000 days, and we have evidence that the companions are white dwarfs. Both point directly to an origin from the mass transfer. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 3:29 p.m. Eastern Time. Quick now. The light from the blue staggerer's companion star is not actually visible. Observations. While the companions haven't been seen directly, their effect on blue staggerers is evident. Each companion pulls gravitationally on its blue staggerer and creates a wobble as it orbits, and this allows astronomers to measure the mass of the companion stars. The WIYN data show that each companion star is about a half the mass of the Sun, which is consistent with a white dwarf. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 21st, 2011, 3:31 p.m. Eastern Time. Quick note. We think we have a good understanding of stellar evolution, but it doesn't predict blue staggerers. People have been trying to explain the origin of blue staggerers since their discovery in 1953, and now we have the detailed observations needed to identify how they were created. I've always enjoyed trying to get to the bottom of the mystery. And good morning, this is Dr. Will, CBMD, PhD. This Monday, October 24, 2011, 12:22 a.m. Eastern Time. Quick note. New evidence for the oldest oxygen-breathing life on land. Science Daily, October 19, 2011. New University of Alberta research shows the first evidence that the first oxygen-breathing bacteria occupied and thrived on land 100 million years earlier than previously thought. The researchers show that the most primitive form of aerobic respiring life on land came into existence 2.48 billion years ago. And good morning, this is Dr. Will, CBMD, PhD. This is Monday, October 24, 2011, 12:23 a.m. Eastern Time. Quick note. The research team led by UNA geomicrobiologists made their find by investigating a link between atmospheric oxygen levels and rising concentrations of chromium in the rock of ancient seabeds. And good morning, this is Dr. Will, CBMD, PhD. This is Monday, October 24, 2011, 12:24 a.m. Eastern Time. Quick note. We suggest that the jump in chromium levels was triggered by the oxidation of mineral pyrite fool's gold on land. And good morning, this is Dr. Will, CBMD, PhD. It is Monday, October 24, 2011, 12:25 a.m. Eastern Time. Quick note. Pyrite oxygen is a simple chemical process driven by two things, bacteria and oxygen. The researchers say this proves that oxygen levels in Earth's atmosphere increased dramatically during that time. And good morning, this is Dr. Will, CBMD, PhD. It is Monday, October 24, 2011, 12:26 a.m. Eastern Time. Quick note. Aerobic bacteria broke down the pyrite, which released acid that dissolved rocks and soils into a cocktail of metals, including chromium. The minerals were then carried to the oceans by runoff of drained water. And good morning, this is Dr. Will, CBMD, PhD. It is Monday, October 24, 2011, 12:26 a.m. Eastern Time. Quick note. Our experimentation of the ancient seabed data shows the chromium levels increased significantly 2.48 billion years ago. This gives us a new date for the Great Oxidation Event, the time when the atmosphere first had oxygen. And good morning, this is Dr. Will, CBMD, PhD. It is Monday, October 24, 2011, 12:27 a.m. Eastern Time. Quick note. The rising levels of atmospheric oxygen fostered the development of a new bacteria species, that following the evolutionary path back to the first oxygen-breathing life form on land. Our ancestors started off in a pool of highly acidic water. And good morning, this is Dr. Will, CBMD, PhD. It is Monday, October 24, 2011, 12:28 a.m. Eastern Time. Quick note. The researchers say the modern analog for the first primitive oxygen-dependent life form on Earth is still with us. The same bacterial life forms are alive and well today, living off pyrite and settling in the highly acidic waste waters of mining sites the world over. And good morning, this is Dr. Will, CBMD, PhD. It is Monday, October 24, 2011, 12:31 a.m. Eastern Time. Quick note. Cooling the warming debate, major new analyst confirms that global warming is real. Science Daily, October 21, 2011. Global warming is real, according to the major study released October 20th, despite issues raised by climate change skeptics and Berkeley Earth's surface. Temperature study finds reliable evidence of a rise in average world land temperature of approximately 1 degree Celsius since the mid-1950s. And good morning, this is Dr. Will, CBMD, PhD. It is Monday, October 24, 2011, 12:32 a.m. Eastern Time. Quick note. Analyzing temperature data from 15 sources, in some cases going as far back as 1800, Berkeley Earth study directly addressed scientific concerns raised by skeptics, including the urban heat island, effect, poor station quality, and the risk of data selection bias. And good morning, this is Dr. Will, CBMD, PhD. It is Monday, October 24, 2011, 12:34 a.m. Eastern Time. Quick note. Brains can support findings that IQ can rise or fall significantly during adolescence. Science Daily, October 20, 2011. IQ, the standard measure of intelligence, can increase or fall significantly during our teenage years. According to the research funded by the Wellcome Trust, and these changes are associated with these changes to the structure of our brains. The findings may have implications for testing and streaming of children during their school years. And good morning, this is Dr. Will, CBMD, PhD. It is Monday, October 24, 2011, 12:35 a.m. Eastern Time. Quick note. Across our lifetime, our intellectual ability is considered to be stable, with intelligent quotient IQ scores taken at one point in time used to predict educational achievement and employment prospects later in life. And good morning, this is Dr. Will, CBMD, PhD. It is Monday, October 24, 2011, 12:38 a.m. Eastern Time. Quick note. Biggest ever study shows no link between mobile phone use and tumors. Science Daily, October 20, 2011. There is no link between long-term use of mobile phones and tumors of the brain or central nervous system. Finds new research published online in the British Medical Journal. And good morning, this is Dr. Will, CBMD, PhD. This Monday, October 24, 2011, 12:40 a.m. Eastern Time. Quick note. In what is described as the largest study on the subject to date, Danish researchers found no evidence that the risk of brain tumors was raised among 358,403 mobile phone subscribers over an 18-year period. And good morning, this is Dr. Will, CBMD, PhD. This Monday, October 24, 2011, 12:40 a.m. Eastern Time. Quick note. The number of people using mobile phones is constantly rising, with more than 5 billion subscriptions worldwide in 2010. This has led to concerns about potential adverse health effects, particularly tumors of the central nervous system. And good morning, this is Dr. Will, CBMD, PhD. This is Monday, October 24, 2011, 12:41 a.m. Eastern Time. Quick note. Previous studies on possible link between phone use and tumors have been inconclusive, particularly on long-term use of mobile phones. Some of this earlier work took the form of case control studies involving small numbers of long-term users and were shown to be prone to error and bias. The International Agency for Research on Cancer, IARC, recently classified radiofrequency electromagnetic fields as emitted by mobile phones as possibly carcinogenic to humans. And good morning, this is Dr. Will, CBMD, PhD. It is Monday, October 24, 2011, 12:42 a.m. Eastern Time. Quick note. The only cohort study investigating mobile phone use and cancer to date, Danish national-wide comparing cancer risk of all 420,095 Danish mobile phone subscribers from 1982 until 1995. with the corresponding risk in the rest of the adult population with follow-up to 1996 and 2002. The study found no evidence of any increased risk of brain or nervous system tumors, or any cancer among mobile phone subscribers. And